So I'd like to start by thanking Michael Kennedy and Peter Kopp and their staffs, particularly Anna and Lauren, who've done an outstanding job of taking care of me and organizing this. They really put a lot of work into it. I'd like to thank you for your interest in this topic and for making time to come and listen to this, because one of the things that happens when science gets politicized is that truth sometimes gets lost in the balance, and claims on both sides can be misleading. And in both the federal debate that has occurred over stem cell research, as well as the debates in various states, including Illinois, there sometimes have been claims on the pro side that have exaggerated the potential benefits, and often misleading claims on the counter side as well, to try to muddy the waters in trying to reduce support for stem cell research. So what I want to do today is talk both about adult stem cell research and some of the cool things that we've learned as a result of studying adult stem cells, as well as embryonic stem cell research. So I'm going to try to tell you three stories today. One is about stem cells and cancer. Second is about stem cells and aging. And the last third of the talk will be about embryonic stem cells and what scientists are really proposing to do, and what some of the misleading information is that you've probably heard. Finally, and I want to note this explicitly, there's going to be time for questions at the end. And I think this is important because a lot of people end up spending a lot of time reading newspaper articles about stem cells and come to these kinds of fora with lots of questions. And oftentimes that's the part of these kinds of opportunities that can be most valuable to people. So I do want to make sure we make good use of it. So I'm going to try to be disciplined about finishing in time to leave time for questions, and I hope that you won't be shy about asking them. So let's see. So academic scientists are sometimes, like this guy, accused of living in an ivory tower. The caption here is "Two barbarians and a professor of barbarian studies." But I have to say that living in the world of stem cell biology, I feel like we're living in the trenches. Because not only are these public policy debates going on that people like me, spend a lot of time federally and in the state talking about these issues, and I appreciate the opportunity to talk to you guys, in part because it's an opportunity to really explain what the scientists really want to do and what's really at stake, and what kinds of issues are in the balance. But also because invariably, whether I'm testifying before Congress or whether I'm in a situation like this, people are here who have juvenile diabetes, who have spinal cord injuries, who have other kinds of diseases, who believe that stem cell research represents their best hope. Now, in some cases, it may be their best hope, and in other cases, maybe it's not their best hope. But the fact that so many people, you can't stand in my shoes without feeling a sense of urgency about these things, because every week I get emails from people saying, my husband was diagnosed with this. The doctors say there's nothing we can do. Do you have any clinical trials that you're planning at the University of Michigan or elsewhere? Is there anything you can do? The people that do the kind of work that people in my laboratory do, what we would like to do more than anything else in the whole world is cure something that couldn't be cured otherwise. And people in my lab are working around the clock trying to discover new cures. But the stuff that was easy to cure has been cured. And the stuff that we continue to work on is really hard to cure. And there are many different problems out there, and we're early in this enterprise. So the exciting thing about stem cell research is that it creates opportunities that don't exist otherwise. And I'm going to try to illustrate some of those things that have emerged from our work in this area. But we owe it to those people, I think, no matter what the probability is. Maybe in some cases there's a 1% chance of success. Maybe in other cases there's a 10% chance. Maybe in other cases there's a 50% chance. But whether it's a remote chance or whether it's likely that we're successful, and no matter how people disagree about that, the fact is that we have to do everything we can to do more for those who are suffering and for whom we can't cure them now. So there are many different kinds of stem cells. That's the first thing that you have to understand. There are embryonic stem cells. See, now I don't know how all these buttons work. I think these slides are randomly moving, and I can't tell whether the computer's just getting bored and I'm spending too much time on each slide. So there are many different kinds of stem cells. There are embryonic stem cells, which only exist at the earliest stages of embryonic development, but which have the capacity to make every cell type in the body. Even though these cells in vivo only exist at the earliest stages of embryonic development, literally within days of fertilization, we can propagate them in culture as lines. So when you hear about the NIH-approved lines or the lines that are approved for federal funding, what they're talking about, on the president's approved list, there are 21 embryonic stem cell lines that were created prior to September 2001. Those lines arise from 21 different embryos. So that's the extent of the genetic diversity that those lines represent. So that is one embryo, one line. So once we create lines, those lines can be propagated indefinitely. And so we can continue to use them over and over again for experiments. There are also tissue-specific stem cells, which in the newspaper are often referred to as adult stem cells. Now, adult is a bit of a misnomer because these tissue-specific stem cells exist both in fetal tissues as well as in adult tissues. But there are many different types of these tissue-specific stem cells. There are blood-forming stem cells that exist in blood-forming tissues throughout life. So all of you have blood-forming stem cells in your bone marrow that make new blood cells every day. And you need those stem cells to make new blood cells. And if those stem cells suddenly went away, you would die within weeks of hematopoietic failure. You would be immunocompromised. You wouldn't be able to fight infections. You wouldn't be able to clot your blood, and you'd die of bleeding and infection. You have stem cells in your nervous system, particularly in your brain. I'll talk a little bit later about how there's a population of stem cells in the forebrain that makes new brain cells every day, including new neurons. There are stem cells in your muscles that are responsible for regenerating muscles when your muscles need to regenerate. Stem cells in various epithelia, such as in the wall of the gut, in breast, in skin, in all kinds of places. So there are many different kinds of adult stem cells. And the thing that distinguishes these adult stem cells from these embryonic stem cells is whereas the embryonic stem cells can make every cell type in your body, the adult stem cells are already partially specialized. So blood-forming stem cells can make all types of blood cells, but they can't make new cells in your brain or your liver or your muscle. You need to have those other types of stem cells to make different cell types in different tissues. So why study stem cells? Well, we have lots of reasons. Stem cell biology encompasses fundamental scientific questions at the forefront of cell and developmental biology, questions that have the potential to change the future of medicine. Stem cells offer the opportunity to better understand the origin of disease, because whether you're thinking about cancer or birth defects, these diseases often involve things that go wrong either in the stem cells or in the mechanisms that normally regulate stem cell function. We have the opportunity to perform cellular therapies. That's the tip of the iceberg. So you often hear about Parkinson's disease and juvenile diabetes, where the idea is maybe we could cure these diseases by replacing the cells that go missing in the disease. I'll talk more about that later. But the kind of application that's probably even more important than that, which you hear less about, is in terms of drug therapy, where we may have an opportunity to change the way we discover drugs by using stem cells, by screening new drug candidates against embryonic stem cells. So when President Reagan's family talks about how important embryonic stem cell research is for the future of Alzheimer's disease, that's what they're talking about, the opportunity to discover new drugs that may be more effective in treating Alzheimer's disease. Now, of course, with all of these reasons, this charges up scientists to answer questions that couldn't be answered before about clinicians to try to treat people differently, but it doesn't necessarily charge up the politicians to support expanded embryonic stem cell research. And in this cartoon, it shows two senators coming down the steps of Congress, and one of them is saying, of course, it would be a different story entirely if we could extract crude oil from stem cells. So improving the way we treat cancer biology. I'm going to tell you a story about cancer because one of the main interests in my laboratory is to understand the mechanisms by which stem cells renew themselves. So stem cells persist throughout adult life in a variety of different tissues. In blood-forming tissues, you have stem cells when you're born, and they persist all the way throughout adult life. In your skin, in your brain, in your muscles, in all of these places. And the way in which stem cells persist throughout life is by undergoing what's called self-renewing divisions. That is, stem cells divide to make more stem cells and thereby perpetuate themselves. And in that way, they're different from most cells in the body, which are not able to perpetuate themselves. So it turns out that the process by which stem cells undergo these self-renewing divisions is hijacked by cancer. You hear about mutations or genetic defects that cause cancer. Invariably, these genetic defects inappropriately activate the mechanisms that normal stem cells use to self-renew. And so in that sense, cancer can be considered a disease of dysregulated self-renewal, where stem cells normally self-renew in a highly regulated manner, with some genes that promote the process, other genes that inhibit the process, and that are present in dynamic equilibrium. That equilibrium gets out of balance in cancer, where the accelerator gets stuck on and the cells self-renew out of control. And so in my laboratory, when we study the mechanisms that stem cells use to self-renew, we invariably not only learn something about normal development and how we can enhance regeneration in the tissue, but also how cancer cells get carried away and hijack this process. And thereby, we identify new ways of trying to intervene therapeutically in cancer. So one of the things that we and others have learned in recent years is changing the way that we think about cancer. For a very long time, we've recognized that tumors are packed with very different kinds of cells. Not every cancer cell looks the same. But the idea was, despite this heterogeneity, that many different cancer cells had the capacity to proliferate extensively and form new tumors. Because it was believed that even a single cancer cell could kill you, the modern therapies that we have for cancer involve eradicating every cancer cell. So our first choice is to chop out the tumor if we possibly can. That's a pretty successful therapy. Our second choice, once we have disseminated metastatic disease, is to use these broadly toxic chemotherapies with the hope that we'll kill the cancer before we kill the patient. But of course, these approaches are so toxic that oftentimes we do kill the patient before we're able to cure the cancer. In contrast, in a number of important cancers, including leukemias and breast cancer and colon cancer and various brain cancers, a very different model of cancer has emerged in recent years called the cancer stem cell model, where the idea is that in a normal tissue, we have a few stem cells that have the capacity to proliferate extensively, and a large number of other cells that arise from the stem cells and that have a very limited ability to divide. So most of the cells in your blood, they arise from rare blood-forming stem cells in your bone marrow, but once they've matured in your blood, they have a limited ability to proliferate. The same thing turns out to be true in cancer, that cancers, at least some types of cancer, are driven by infrequent cancer stem cells, which retain the capacity to proliferate extensively and to form new tumors. But just as a normal stem cell gives rise to diverse types of specialized cells with a limited ability to proliferate, so do cancer stem cells, giving rise to large numbers of cancer cells that have a limited ability to divide. So, in fact, in testicular cancer, this is the one solid malignancy that we can cure pretty effectively even after it becomes metastatic. We've known for about 30 or 40 years that there are rare testicular cancer cells that are undifferentiated, and those are the ones that we really have to kill. And they give rise to all kinds of crazy other cells, hair and teeth and neurons and crazy stuff that loses its ability to divide in becoming specialized. It turns out that that's also true of other cancers like the ones that I've listed on here. And so if that's true, what this really means is that rather than using broadly toxic approaches to try to kill all of these cells, we really just have to kill the cancer stem cells. The trick is, how do we recognize those cells? So the idea that has emerged here is that over the past 30, 40 years, we've not understanding these differences among cancer cells, we have screened for new cancer therapies based on their ability to shrink tumors. And an idea, and this has not been proven yet, this is an idea, the idea in the field is that because most of the cells within a tumor are these partially specialized cancer cells that have a limited ability to divide, in screening for drugs that will shrink a tumor, we've inadvertently selected for drugs that kill off these cancer cells with limited proliferative potential. So we have all kinds of drugs that can shrink tumors down to the point where we can no longer see them radiologically. And yet we know, in the context of certain cancers, that 99% of those patients, after we've shrunk their tumors back to the point of no longer being able to detect them, we know that 99% of those patients will go on to die of metastatic disease. The idea is that's because we're leaving the cancer stem cells behind. And that instead of screening for drugs that just shrink a tumor, we should be screening for drugs that kill these cancer stem cells. So that even if we don't shrink the tumor, if we kill off the cancer stem cells, we'll convert a malignancy to what's effectively a benign tumor with a limited ability to proliferate. And the one success that we've had in metastatic solid cancer is with cisplatin compounds in testicular cancer, where this is exactly what they do. And you can leave behind the hair and the teeth and the neurons, and they will almost never come back and kill the patient. So now the challenge is to try to apply these approaches to other cancers where these differences have been less apparent. So now, one cancer where this is particularly important is in the context of leukemia, because what we would really like to do is to develop approaches to kill the leukemic stem cells without harming the normal stem cells. And that's a really difficult thing to do because the leukemic stem cells are using all of the same machinery to proliferate as the normal blood-forming stem cells are. And so it's a hard problem to try to find drugs that will kill the leukemic stem cells without harming the normal blood-forming stem cells. And here's why. There are certain genes, and I don't want to get too technical here, called proto-oncogenes that are growth-promoting genes. These are genes that when a cell turns them on, they cause that cell to divide. So these genes that include kind of a lot of pathways that are well-known and frequently implicated in cancer, one of which is a gene called BMI1. I'm only mentioning it because I'm going to mention it briefly later. I'm going to mention this again. All you need to know is that this is one of these growth-promoting genes. And so oftentimes you see this overexpressed in cancer cells, and that's the way that the cancer cells divide out of control. But these proto-oncogenes not only promote the proliferation of cancer cells, they also promote the proliferation of the normal stem cells. Now, these proto-oncogenes are normally balanced against tumor suppressor genes, which inhibit growth. So you have growth-promoting signals and growth-inhibiting signals. And these growth-inhibiting signals not only inhibit cancer cell proliferation, but also inhibit normal stem cell self-renewal. And so under normal conditions in normal tissues, the growth-promoting signals are balanced with the growth-inhibiting signals so that everything stays kind of at steady state. And in cancer, you either amplify these signals or you delete these signals. A couple of genes that are important tumor suppressor genes that I'm going to again mention later and try not to get too technical are INK4A, ARF, and PTEN. These genes are powerful growth-inhibiting signals that are frequently deleted by cancer cells. Cancer cells just eliminate them from the genome and thereby don't have this set of brakes anymore on the process and will divide out of control. So the problem here is if all the same mechanisms are regulating both the normal stem cells and the cancer stem cells, how do we kill the cancer stem cells without killing the normal stem cells? In an ideal world, what you'd like is to identify a rare example of